So the next structure which I'm going to discuss in our immunology anatomy section is the lymph node. And first we discussed a secondary organ of immune system, spleen, and now we discuss lymph node. Lymph node. So lymph node is an encapsulated structure, and here we will draw. There is an efferent. It has many afferents and only one efferent, lymphatics. So this is, for example, a one section of a lymph node having the one afferent. It is encapsulated. So this is the capsule. It is having tubercules like spleen. This is the capsule. This is the tubercular structure, and in the tubercle this is a cortical area. So in cortex we have a primary follicle, the growing primary follicle, which turns into secondary follicle. This is the primary follicle. This is the secondary follicle having the germinal center. Secondary follicle having the germinal center. Then we have the paracorticular area. Next to the cortex, into the medulla, we have paracorticular area, and then we have medulla and medullary sinuses and medullary cords. So this is just a one section of the lymph node, and over here we have the only one efferent lymphatics. So lymph node has many afferents, only one efferent. Function of lymph node. Lymph node function as a filtration organ. It filters macrophages, B cells and T cells. And also it is an immune organ, so it is related to immune response. Then we have in our corticular area we have a follicle known as primary follicle and secondary follicle. Primary follicle is dense, secondary follicle is pale, primary follicle is dormant, as it is represent as a primary precursor organ which can convert it into secondary follicle in case of any infection. And we have secondary follicle having a pale in color and having germinal center. And if this follicular area, this follicular area is underdeveloped, then we will have an immunodeficiency condition known as B cell disorder or Bruton A gamma globulinemia, which we will discuss in our next lecture of immunodeficiencies. So in the next part we have this paracorticular area, and in this paracorticular area we have lymphatic blood supply, lymphatic artery, and the lymphatic vein. So this vascular structure over here in the paracorticular area is highly endothelial venule, through which the B cells and the T cells pass into the bloodstream. This is venule. So paracorticular area consists T cells, Also dendritic cells, which is a part of APC, antigen presenting cell, dendritic cells. Paracortical area has high endothelial venules, postcapillary venules, through which T cell pass into the bloodstream for immune response. And if we have any extreme viral infection, let's suppose we have EBV, then this paracortical part hyper goes under hyperplasia, and we have lymphadenopathies. Next, we have this medullary part. This medullary part consisting of two things: medullary sinuses, consisting of macrophages and reticular cells. Sinuses connects efferent lymphatics to the bloodstream, and we have the cords. Cords has plasma cells and lymphocytes. So this is a general anatomy and physiology of a lymph node, having different parts of cells. It can filter macrophages, B cell, T cell, so filtration, and then immune response. Then we have primary and secondary follicle in the cortical area. Primary and secondary. Primary has dense and dormant follicle, and secondary is active or pale in color, having germinal center.